Hello. Hi, everyone. Hope you're hope all you're doing good. So today I'm starting with the chapter number 11 of class 11th biology, which is photosynthesis in higher plants. So myself, Mahendra Singh. So I have done PhD in genetics and plant breeding, and that's PhD in agriculture. And today I'm going to briefly tell about the first part of this photosynthesis early experiments and how they find out the equation of photosynthesis and what are the experiments or what are the preliminary things they did before concluding for the photosynthesis equation. So I will just go by line to line. So it's good to go by line to line and explain the important stuffs, important things, what is written in the NCERT textbook. So first I will start reading. So all animals, including human beings, depend on plants for their food. So the first introduction part is just say that the green plants are autotrophs because they can do the photosynthesis. So just they can do the photosynthesis, they can produce their own food. That's why it is called as autotrophs. Can go here, autotrophs, and it's a physiochemical process. I think this physical, I don't know, it's a physiochemical. It should be physiochemical process. Why is a physiochemical process? Photosynthesis is a physiological and chemical process. Physio means physiological. So physiological process includes because photosynthesis to happen photosynthesis we do require light, we do require water, we do require oxygen. So the plant absorbs light, take water, and take oxygen from the external environment through physiological process. So all three, the intake of water, absorption of light, and intake of oxygen through stomatal pores is physiological process. What are the chemical process? The chemical process we have is fixation of carbon dioxide to glucose and breaking down of water and all other reactions which involved of different elements and chemicals are called chemical process. That's why it's called as physiochemical process. So coming to 11.1, so they have done a lot of simple experiments, very simple in the 17th century and the 18th century. It's a very simple experiment to prove that what plants are really producing and what plants need to produce oxygen or to fix carbon to glucose. Okay, so this part is just an introduction part. I'll just go let me go for this. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black pepper and exposed to light. So there's a starch iodine test, where when you put iodine is mostly in orange color. When you put iodine on starch, if the compound is if the substance is having starch, then it will turn into blue to black color. If the substance that doesn't has starch, it doesn't change into color. So this experiment you already know, where they took the leaf and they covered the part of the leaf with the black paper, and other part of the leaf they exposed to the sunlight. When they did the iodine test, when they put the iodine on both exposed part of the leaf and covered part of the leaf. Only the exposed part of the leaf turn into blue and black. That means the only exposed part is producing starch. So to produce starch, light is required. That's what it's all saying. This one. So another experiment. This is all. I think it's all been well discussed in previous chapters, but it has been given in this chapter. I just want to discuss that also. So another experiment is CO2 required for photosynthesis. That's carbon dioxide required for photosynthesis. Thus is a KOH soaked cotton experiment where they took a test tube and they put a leaf, a single leaf like this. Maybe it's a rice leaf or wheat leaf or any leaf. They put a leaf and they covered here a cotton, soaked cotton, which is soaked with KOH. KOH. The special property of KOH is it can absorb carbon dioxide. So it has a leaves here. The half leaf is exposed to the air, and half leaf is in the test tube. For the test tube, it is plugged with cotton, which is soaked with KOH. So later they kept this whole setup in the light. They kept this whole setup in the light, and after some time they checked for the starch, the same iodine test. So it was very exciting to see that only the exposed part of the leaf turned into blue to black, and the unexposed part, the part which is inside the inside the test tube, didn't change the color. So what does it really indicates? Can you give people guess? The exposed part, which is getting something from the external environment and which is making the starch to produce. But what element? That's how the KOH soaked cotton thing comes into the picture. The KOH absorbs the carbon dioxide, but it doesn't absorbs oxygen. So inside the tube, the oxygen is flowing, but only the carbon dioxide is restricted. The carbon dioxide is absorbed by KOH. So it didn't turn into the blue or black color. So it indicates carbon dioxide is essential for photosynthesis. So hope you understood these two concepts. So coming on to the early experiments, that's 11.2. So I told you that these experiments are very, very simple, but it is a lot of informative. The conclusions were tremendous at the time. So in the 1770, the Joseph Priestley, he performed a series of experiments that revealed that role of air in growth of green plants. So what he did, he took a jar and he light up the candle inside that and he kept the mouse. So he closed the burning candle and the mouse inside a jar and he kept it for some time. After some time he observed that the light was off, the candle was off, and the mouse was dead. But when he did the same setup with a mint plant inside, so after some time he saw that the candle was still burning and the mouse was still alive. So what he concluded, the only difference is the mint plant here. So he concluded that a burning candle or an animal that breathe the air both somehow damage the air. From this first A and B concluded, because the burning candle and the breathing animal, this is the breathing animal, is somehow damaging the air. That's why this both the burning candle was off and the mouse were dead. But when placed a mint plant inside the same bell jar, he found that mouse stayed alive and the candle continued to burn. So this is really exciting. The only difference is the mint plants. So Priestley hypothesis as follows: plants restore to the air whatever breathing animals and burning candles remove. So we all know that breathing animals and for the burning candle we required oxygen. So plants is restoring oxygen which is consumed by the breathing animal, that's a mouse, and the burning candle. So what it indicates the Joseph Priestley discovered that plants releases oxygen, so which we need, all animals need to breathe. So Joseph Priestley is the person who gave the first evidence of O2 is released by the green plants. So can you imagine how Priestley would have conducted the experiment using a candle and a plant? Yeah, the same how which was mentioned here. Remember, you would need to rekindle the candle to test whether it burns after a few days. So these are simple questions you can always fond of, and you can always think about this. So going on to the next part, using a similar setup as the one used by Priestley, but by placing it once in the dark, once in the sunlight. The same setup what he has used here. So, but he placed it once in sunlight, once in dark. The same one. Jan Ingenhousz showed that sunlight is essential for the plant processes that somehow purifies the air fouled by burning candles or breathing animals. Ingenhousz is an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. So what he did simply, he took the same one, and one he kept in the light, one he kept in the dark condition, and he found that the one which is kept in the light condition was the mouse were alive, and the candle was burning. The one which is kept in the dark condition, the mouse was dead, and the candle was not burning. So what it indicates, to release oxygen, plants need sunlight. Only in the presence of sunlight, the plants can release oxygen. This is how the mouse and the lights and the candle was still alive. So this is given by Jan Ingenhousz. And even like in the aquatic plants, in the presence of sunlight, the small bubbles were formed around the green parts of the plant, while in the dark they did not. So what it did, it took the green plant, the aquatic plant, which is aquatic plants means the plants which can grow inside the water. So he kept that, he took a beaker, he took a pot which has water and which is having aquatic plant. He kept in the light, he kept in the one pot with the plant he kept in the light, another pot with the plant he kept in the dark conditions, and he did see only under the light conditions he saw the bubbles coming out. So Jan Ingenhousz was the person who proved light is required to evolve oxygen from the green plants.